Hi students, welcome to ANT Science and Pharma. As we already spoke about the basics of carbohydrates, the definition, now we are going to see in detail about carbohydrates. Now, now coming back to the definition of carbohydrates, carbohydrates are organic compounds that has aldehyde group or ketone group and polyhydroxy groups. That is what we have discussed. Now, see, the same thing is given here. Carbohydrates are polyhydroxy, aldehyde or ketone. Aldehyde is CHO, ketone is C double bond O. So all the organic compounds that contain polyhydroxy groups, OH groups are there, many OH groups are there, aldehyde group along with an aldehyde or a ketone group. Those are called as carbohydrates. Okay. So this is your general structure of carbohydrate, that is CN H2O N. Okay, usually we write it otherwise as CN H2N ON. So usually this method will be confusing for some people, like H2O means water, know how it is. Yes, some reactions will lead to water only, removal of OH will lead to water only in some reactions, but consider this as a standard thing. CN H2N ON. N is number of carbon atoms, that you have to keep in mind. If in a molecule there are 6 carbon atoms, hydrogen will be 2 into 6, that is 12. Oxygen will be 6. Correct. In this kind of compound, it can be a CHO group or C double bond O group, anything. Okay, but both are considered as carbohydrates only. Now, so how the carbohydrates are synthesized? You all know plants synthesize carbohydrates from photosynthesis, correct? From CO2 and water. From this both plants synthesize carbohydrates using sunlight, correct? No, sunlight. And in human beings or in animals, what happens is carbohydrates will get broken down into CO2 and H2O. So this is a reversible process in our environment. Okay, so carbon dioxide and water combine to form carbohydrates, carbs. So in animals, what happens is we consume those carbohydrates to generate some energy. Right? Here, energy is uh, endergogenic in the form of sunlight. Energy is intaken in the form of sunlight. Here, energy is exergogenic in the form of ATPs. In animals and human beings, carbohydrates are broken down to form carbon dioxide and water to give ATPs. ATPs are the sources of energy. Now, coming to the classification of carbohydrates. Carbohydrates are usually classified based on the functional group, presence of aldehyde group or ketone group. These are called as aldoses. Aldo. These are called as ketoses. So, irrespective of number of carbon atoms, if that organic molecule, if that carbohydrate contains aldehyde group, those are termed as aldoses. If the carbohydrate contains ketone molecule, that is ketone functional group, those are called as ketoses. Okay, based on the number of carbon atoms, also the carbohydrates are classified into numerous ways. But based on the functional group, they are classified as aldoses and ketones. These are the classic examples of aldose and ketones, glucose and fructose. Okay, see here, this is glucose molecule, CHO is present. This is fructose molecule, CO is present. What's the here aldehyde is there. If the first carbon can't we give CHO, it is not possible. If it gives, it becomes an aldehyde group. So as usual, we know ketone group always it is present in the middle of the molecule. Now I told you, based on the number of carbon atoms present in the carbohydrate, based on the number of carbon atoms present in the carbohydrate, They are classified into this many types: triose, tetrose, pentose, hexose, and heptose. This triose contains three carbon atoms. Three carbon atoms. So, if the molecule contains three carbon atoms, how many hydrogens will be there? Six. How many oxygens? Three. Heteros. How many carbons? Four. How many hydrogens? Eight. How many oxygens? Four. How many carbons? Five, 10, and 5. Hexose. C6, H12, O6. Heptose. C7, H14, O7. We have already seen the examples of aldohexose. What is that? Glucose. See here, aldose, glucose. It contains 6 carbon atoms. 1, 2, 3, 4, 5, 6. Fructose contain 1, 2. See, the here plus is there. That means one carbon is there. One carbon is there. One carbon is there. So 1, 2, 3, 4, 5, 6 carbon atoms. So these are hexoses. Aldotriose. Usually triose means it does not differentiate between aldehyde or ketone functional group. If you want to differentiate, it is called as aldotriose, ketotriose. Aldotetrose, ketotetrose means all the compounds contain aldehyde group here. All the compounds contain ketone functional group. Don't confuse. This is not the name of the compound. This is the classification of the compound only. Aldotriose is not a compound. Aldohexose is not a compound. These are the classification only. So, for example, aldohexose is glucose. All the examples I will tell now. Benzaldehyde is there, erythrose is there, arabinose is there, xylose is there, glucose. Like that, various examples are there. But these are the classification terms only. Right. Now we will see the examples of what are this. Aldose means it will have aldehyde group. So this contain 1, 2, and 3 with aldehyde group. That is why it is called as glyceraldehyde. Glyceraldehyde is the example of C3 containing carbohydrate with aldehyde group. That is glyceraldehyde. Now we have 4 C4 1, 2, 3, 4. This is erythrose. And this is triose. What is the difference? The orientation of OH is different. Okay, this is erythrose and that is triose. And the molecules coming from erythrose are C5 carbon atoms. C5. All these things are C5, but aldehyde are there commonly for everything. Examples will be ribose. See, it is just addition of another carbon and OH. See, 1, 2 are there, 1, 2, 3 are there. Erythrose and ribose. And next molecule, only difference will be the orientation of this OH group. Orientation, if it is changed completely, it is called as arabinose. Okay, from triose. So middle carbon is changing now. So middle carbon is changing. Here second carbon is changing. You remember? Here third carbon orientation is changed. That is xylose. From ribose to xylose, what is the difference? Third carbon is different. If it is second carbon, it is called as arabinose. If it is third carbon, it is called as xylose. If it is fourth carbon, 1, 2, 3, fourth carbon. If it is fourth carbon, it is called as lysose. So all the hydroxy groups are aligned to one side. It is called as ribose. If second hydroxy group on second carbon has aligned to the left side, it is called as arabinose. If third hydroxy, it is called as xylose. If fourth hydroxy group is aligned on the left side, it is called as lysose. Okay. Now we will see 6 carbon C6 with aldehyde structure. C6 with aldehyde structure. And you might get, by seeing the structure, you might get one doubt. Here C double bond O is there. No. Is that not a ketone group? No, because you have C. This is CHO group, that is aldehyde group. So don't get confused. These are all aldehyde groups only. Now, direct descendant from ribose. All the hydroxy groups on the left side, C6 carbon, that is called as allose. allose. This is allose. When second carbon hydroxy is shifted, it is called as altrose. Allose, altrose. When third carbon is shifted, it is called as glucose. See here, third carbon is shifted. That is glucose. Okay. Next we will go for uh, two carbon atoms. I told you mannose in the previous exam. In the previous slide, I told you mannose. Two carbons are there. Two carbons are there. It will go on forever. And galactose also, I think we discussed. See here, middle two are there. Okay, so this is the classification of aldoses. These are all aldoses. C3 containing glyceraldehyde, C4 containing erythrose and triose, C5 containing ribose, arabinose, xylose and lixose, C6 containing allose, altrose, glucose, mannose, galactose, lactose, etc. Okay. Now ketones. Same goes with the ketones also. Dihydroxyacetone, C3. C3 is dihydroxyacetone. So all the are aligned to the same side. Erythrose, where erythrose we have seen. Here you see three are aligned to the same side. Ribulose, their ribose we have seen. Okay, from here third carbon we have seen shifting to here. It is called as xylose. Here xylulose. See third carbon xylulose. Okay, and the same goes with the C6 also. Siccose, siccose, fructose, okay, sorbose and lagatose. Okay, this is similar to the glucose. Glucose also same carbon atom, we have hydroxy group on this side. If all the hydroxy on the same side, if it is aldose, it is called as allose. You remember allose. If this has gone here, altrose. Okay, here siccose. All the hydroxy groups are aligned on the same side. That is siccose. Okay, so now, so previously we have got monosaccharide, oligosaccharide and polysaccharide will be there. Trisaccharide, polysaccharide, polysaccharide. So what are these polysaccharides? What are these monosaccharides? What are these trisaccharides, polysaccharides? Now, see, we have seen C3, C4, C5, C6, C7 containing carbohydrates. Correct? These are single molecule. Single molecule. So C3 means if it is having aldose, it is called as aldotriose. Correct? So this is one molecule. One molecule. One molecule of carbohydrate or one molecule of sugar you can say. Okay? Or otherwise it is called as monomer. 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 Okay. Now in monosaccharides, only one molecule of carbohydrate is present. Either C3 or C4 or C5 or C6 or C7 means number of carbon atoms. So either one of those is present in a either one of them is present. That is called as a monosaccharide. Mono means one. Correct. It is called as monomer. Only one is present. So in the monosaccharide, when you want to hydrolyze the monosaccharide, it does not form any simpler form of the sugar. So it is the final form. That means it has only one molecule. It does not form any other molecule by hydrolysis. That is monosaccharide. Now, what are oligosaccharides? Here, 2 to 10 monosaccharide means oligosaccharide. They told 2 to 10 monosaccharides. That means, for example, of glucose, we can say fructose. One glucose and one fructose is there. That is called as sucrose. Glucose contain C6. Fructose contain C6. So how many C should be there? C12. Okay, that is fructose. Now, if glucose powder is taken in one bowl and fructose is poured into one bowl, when you mix it, it does not become sucrose. A physical mixture of two monosaccharides does not term it as disaccharide. When two molecules are present, it is called as disaccharide. Here you are told 2 to 10 monosaccharides. No. So if two molecules are present, it is disaccharide. If three molecules are present, it is trisaccharide. If more number of molecules are present, it is polysaccharide. Understood? Now, if you pour glucose and fructose in a bowl and mix together, it does not form sucrose. Then how sucrose is formed with the uh, uh, mixture of glucose and fructose means these are linked using glycosidic linkage. Glycosidic linkage. See, you have Howard projections like this, Howard projections like this, OH, OH. So one of those hydroxy groups, see if any other molecule is here, any other in this sense glucose or fructose. In this current example, OH is there, OH is there. So here CH2 OH is there. So if this elimination of water molecule H2O, so it will join together with the help of O. It will join together with the help of O. It is joining with the help of O. This is called as glycosidic linkage. As you have seen, two molecules of monosaccharides are linked using glycosidic linkage to form a disaccharide. In this example, sucrose is a disaccharide. Upon hydrolysis, that is giving glucose and fructose, two molecules of one glucose and one fructose, it is forming. Okay, so this entire structure is called as sucrose. Okay, similarly, maltose is another disaccharide. When hydrolyzed, it will give two glucose molecules, two separate glucose molecules. Understood? So now, similarly to disaccharides, we have trisaccharides. The name itself, the name itself indicates tri means three, not C3. It is three monosaccharides. When trisaccharides are hydrolyzed, it gives three monosaccharides. Example, raffinose. When raffinose is hydrolyzed, we get one glucose molecule, one fructose molecule, and one galactose molecules. Okay. So now we have polysaccharides. Polysaccharides means each polysaccharide molecule when hydrolyzed will give more number of monosaccharide units. So one polysaccharide will be a combination of different kinds of monosaccharide units or same kind of monosaccharide units. Those are bonded together. Now here polysaccharide is starch. Starch when hydrolyzed will give more number of glucose molecules. Correct. From that only we are getting energy. When we are eating corn, potato, wheat, rice, we get starch. When starch is broken down inside our stomach, hydrolysis, it will lead to glucose. Glucose will go into glycolysis pathway and will generate ATP. You remember that? ATP. Correct. Now, starch will contain two types of molecules, amylose and amylopectin. Amylopectin. Amylose and amylopectin. So now when glucose molecules are linking together with the glycosidic linkage, two types of bonds will happen. Glucose binds to glucose, binds to glucose, binds to glucose, and so on and so on. This is one kind of linkage. Now other kind of linkage, glucose, glucose, down, glucose, glucose, down, glucose, glucose. So here we have glucose to glucose 1,4 linkage. 1,4 linkage based on the positions, based on the alignment. I will tell you what is 1,4 linkage, but as of now you remember this. Glucose is linked to other glucose. In all the cases, if glucose and glucose are linked via 1,4 linkage, it is called as amylose. When glucose and glucose are linked as 1,4 and also 1,6. 1,4 and 1,6. If the both linkages are present, it is called as amylopectin. So you all know amylose will be a straight chain compound. Amylopectin will be branched chain compound. So this is less rigid, it is soft, less rigid, and this is very rigid. Easily digestible, it is hard to digest because it is having all the same linkages, 1,4 linkages, easily digestible. It is little hard, so it also has 1,4 along with 1,6 linkages, that is the polysaccharide. Now we have one peculiar thing with the carbohydrates called as DL rotations. Based on the rotation or the confrontation of OH groups that are present in the molecule, the D form or L form is decided. See, now we are seeing glyceraldehyde. Usually this we will show glyceraldehyde as COH. That is how we will show glyceraldehyde. Okay, the position is inversed here, no problem. So this is glyceraldehyde. When the OH group is rotated this way on the right side, it is called as D form. D means dextro. D means dextro. When the OH is on the left side, it is levo. L means levo, not left. Okay, dextro and levo form. What is the importance of dextro and levo forms are is there are few molecules, few configuration only will be active form, other configuration will be inactive form. For example, cetirizine, cetirizine HCl, that is antihistamine drug, correct? Antihistamine drug. So in cetirizine, L form is active, levocetirizine is active form, dextro is inactive form. Okay, because it does not perfectly fit into the receptor. That is the reason levoforms are levoforms and dextroform configuration is very much important to determine the pharmacological activity. Understood? Okay, so when the one carbon, only one carbon alignment is changing, one carbon configuration is changing. See here, configuration is changing only at one carbon and remaining everything is same to same. Correct? Only at one carbon the arrangement is changing. So these are called as epimers. These kinds of compounds are called as epimers. When the configuration only at one carbon varies, those are called as epimers. And it is happening at the second carbon. So these are called as C2 epimers. C2 epimers. Glucose and mannose are C2 epimers. When epimerization is happening at the third carbon, okay, this is called as idios and talos. These are C3 epimers. C3 epimers. You don't need to remember everything as such, but try to understand it. Once you understand, you will not forget it forever. Okay. Now, at the fourth carbon, if it is happening, these are called as C4 epimers. This is called as fructose. Remember, ketone group is a fructose and tagatose. Fructose and tagatose. So, any whatever the molecule it is, if one configuration of one carbon atom is changing, that is called as epimer, and the phenomenon is called as epimerization. And based on which carbon the epimerization is happening, those are termed as C2 epimer, C3 epimer, C4 epimer, and these are your classic examples.